A very good day to all of you. So today we are going to discuss regarding interpretation of genetic variants. So it is very important in today's age when a large number of techniques like exome sequencing, genome sequencing are available, where a large number of variants are detected. However, it is very important that we need to interpret whether a variant is disease causing or not. So why do we need to interpret a variant? So if you look here for this picture, this looks like a very harmless kind of leaves which are lying here. But if you look closely, then this is actually a lizard which has camouflaged itself to look like a leaf, and it is very dangerous to the insects. On the other hand, if we think of this animal, it's a very dangerous animal, but here you can see that he is a very meek animal and very quietly sitting with this human. So sometimes harmless looking variants can be disease causing, and the classical example of this is this synonymous mutation which leads to a very severe disease, that is progeria. On the other hand, a harmful looking variant can sometimes be a normal polymorphism. So whatever we feel like by looking at the variant, we cannot be sure that that is what this variant is causing. Hence, it is very important to follow a systematic way to identify a particular variant. So let us understand some of the terms that we will be using during this variant interpretation. Variation refers to difference in DNA among different individuals. Mutation is defined as a heritable alteration or change in the genetic material. And polymorphism is defined as inheritance of a trait controlled by a single genetic locus with two alleles, in which the least common allele has a frequency of about 1% or more. Now, we don't prefer to use the word mutation since it has a different connotation, and we will prefer to use the word variation whenever we want to talk about alteration or change in the genetic material. Now, the question comes, can I really do variant interpretation? And this is very, very important because today's biologist also needs to be a computational specialist, and today's computational specialist also needs to understand some of the biological principles. So it is very important that all life science students or all doctors who are involved in interpretation of the exomes or of the NGS based analysis should know how to do a variant interpretation. Although it looks little difficult by looking at all the jargon in variant interpretation, but as we know that we have examples where people say anybody can dance or anybody can sing, and today we hope that after this presentation you also should be able to say that anybody should be able to do variant interpretation. And it's very important, if you follow a systematic method, then variant interpretation can be much easy. So these are the different steps of variant interpretation. We will go stepwise for each variant. If you follow these all 6 steps, then area variant can be interpreted as disease causing or benign. So the 6 steps include, first is information about the location or functional effect of that particular variant. Then it is the frequency of the variant in normal population. Then whether this variant is reported in other cases with similar phenotype. Then pathogenicity prediction for this variant, followed by Mendelian segregation analysis in the family, and functional assay wherever possible. So all these parts of this jigsaw puzzle, when they come together, they lead to a accurate variant interpretation. So let us go point wise, each evidence how we are going to use to do variant interpretation. So the first one is location or functional effect of the variant. So different variants can be classified based on the location within the gene. So you can see a gene has a promoter, then it has the 5' untranslated region, followed by the translation start codon, that is ATG. Then there is a red colored exon, then there is an intron which is not coded, and then an exon, intron, and then the stop codon followed by the 3' UTR region. And each exon and intron, they have this GTAG consensus sequences which are present at the intron exon junction. So if we know this anatomy of the gene, then we can classify a variant based on its location as exonic, intronic, splice site, 3' UTR, 5' UTR, promoter or enhancer region. Or based on its functional effect, we can classify it as missense, nonsense, frame shift, in frame, or silent. also as synonymous or non-synonymous mutations. So let us see these different types of functional classifications and how they can help to interpret the variant. So this is an example of a patient who came with coarse facies, normal intellectual functioning, and he had these X-ray features typical of dysostosis multiplex. So enzyme assay showed that he has deficiency of aryl sulfatase B, so he is having Marothiolemi syndrome, and the mutation analysis showed that there is a point mutation at this particular location. Now this is a missense mutation because it changes aspartic acid to asparagine. Now we need to see how this missense mutation must be leading to disease, and there some in silico analysis can help us, like this variant is present in the catalytic site of this enzyme and it forms calcium bonds with other surrounding amino acids. So it is a very important kind of amino acid, and any change here can lead to disease. Similarly, when there is a nonsense mutation, it can lead to either nonsense mediated decay or not, based on whether it is within this 55 nucleotides from the next intron exon junction or not. So in this particular patient who had a spasticity, we identified a stop codon mutation here. And the stop codon mutation, then you can check whether it causes a nonsense mediated decay or not. So here it is causing a partial nonsense mediated decay. So stop codon mutations are considered to be more likely to be disease causing. Next, we go on to the splice site mutations. So in splice site mutations, we have the GTAG, which are the consensus splice sites. So any mutation in this particular region can lead to altered splicing. So splice site mutation also is considered as a loss of function mutation or a severe mutation. Like in this patient who had Schwartz-Jampel syndrome, we can see that this is the exon here, and here the intron starts. So this is a intronic +5 mutation G2A, and it is predicted to be splice site mutation. And when we did cDNA sequencing, we could see that because of this mutation, exon 37 and skipped, and 36 is followed by 38. So splice site mutation can again be more likely to be disease causing. Similarly, a frame shift mutation which can result from a deletion or duplication can either lead to a premature termination codon or altered amino acid sequence after that particular mutation. So altered frame shift can again lead to a loss of function mutation, which is going to be more severe. So a single base deletion, like in this patient with leukodystrophy, when we did the sequencing, we found that there is a single base deletion here. And because of this single base deletion, instead of the stop codon being at the normal location, early stop codon is coming because of the frame shift and change of amino acid sequence. So there is a stop codon after 38 amino acids, and a smaller protein is formed which can undergo nonsense mediated decay and lead to the disease. So the type of mutation helps us to interpret whether it is likely to be disease causing or not. A missense mutation is less likely compared to a frame shift or a nonsense mutation. So that is one evidence that we are going to use. The other evidence comes from the frequency of the variant in the normal population. So why is this important? So if you look at different humans, we can see that it's only 0.1% of the genome which is different, but it leads to formation of different types of individuals that we see on this earth. So this just 0.1%, which are the polymorphisms present in our genome, lead to differences among each other. Now, this is a very important graph that we need to understand to know how the frequency of variant in population can help us in interpretation. So if you can see here, on the y-axis, we have the effect size, and on the x-axis, we have the frequency in population. So there are certain variants which are common in population and they have very less effects. So these common variants in population which have very less effect will mostly be involved in the multifactorial disorders, whereas these are the variants which are rare in the population, but they have very high effect. And these are the variants which we are mainly interested in identifying in genetic diseases. So we can filter out all these variants which are common in the population and focus only on the rare variants. So typically that's why we will look that any variant which is less than 1% of the population, we will consider it as likely to be pathogenic. If it is more than 1% of the population, then it is very likely to be benign. But we should remember that certain variants which are common in population, like we have carrier frequency of thalassemia, which is up to 3%. So these variants need to be kept in mind while interpreting in this way. And why is this important to know is that in different populations, we'll have different distribution of the variants, and hence each population, we should have the normal population data to interpret the pathogenicity of a variant. So in order to do this, a number of databases have been developed, like the 1000 genome, the exome variant server, the exact, then the GenomeAD, the genome aggregation database, then complete genomics, the greater Middle East variome, the Genome Asia project, and others. So all these consists of thousands of exomes or genomes which have been accumulated from normal individuals, and the frequency in the normal individuals helps us to interpret the variant. So these are the websites of all these databases. You can see the GenomeAD, the Genome Asia, the GME Variome, and the 1000 Genome Project here. Similarly, in the Indian population also, we have a few databases which have been created, which include the SNP-DB from TMC, the Indian Exome Database, CleanDB, the Indie Genomes, and the recent Indian Exome Database, which has been published in 2021. So one of these databases was developed by us in collaboration with 4 different institutes throughout the country. And this database consists about 1,455 exomes. And this is freely available from the website of CDFD. And similarly, there is an effort going on now called as the Genome India, where 10,000 Indian genome population is going to be sequenced. So it is very important that we have an Indian population data to do a correct interpretation. The next databases that we need are the known variant databases. So these are databases which tell us where known variants in earlier patients have been described. So ClinVar is a very important database here where we can check for known variants in diseases. Similarly, UMIM, PubMed, or the paid database, that is the human gene mutation database, also helps us to detect known variants in earlier patients. The third step is pathogenicity prediction. So here the amino acid is checked for its probability of pathogenicity using different mechanisms. So one is using the conservation score, where you look that whether an amino acid is conserved across species or not. So these are different species. You can see here this amino acid is different in different species, whereas this amino acid is same in all the species. So this is a very important amino acid. Any mutation at this amino acid is more likely to be disease causing. Similarly, we look at the physiochemical properties of the amino acid, like whether it is a polar or non-polar, hydrophobic or aliphatic, aromatic, positively charged or negatively charged. Based on this also, we can interpret whether it is likely to be pathogenic or not. And then again, the 3D structure, the alpha helix and the beta pleated sheet and the tertiary structure, these all analysis also help us to predict whether a variant is likely to be disease causing or not. So there are a large number of pathogenicity prediction tools available. More than 100 such tools are available, and commonly used are the SIFT, Polyphen, the CAD score. So for CAD score, we use a score of more than 20 as likely to be disease causing. So these are the websites of these all pathogenicity prediction software. Mutation Tester is a very user-friendly kind of a software. Then CAD and others are also available. The next step and very important step is Mendelian segregation in the family. So here, once we identify a variant in the exome sequencing, and it may be reported as a variant of unknown significance, and now we need to interpret whether this variant is likely to be pathogenic or not. So Mendelian segregation in the family helps us to further classify a heterozygous or a homozygous variant into disease causing or not. So let us see in a heterozygous variant. So when proband has a heterozygous variant and it is classified as a variant of unknown significance. So here we do the segregation analysis in the parent. So we do Sanger sequencing for the same variant in the parent. So when both parents do not have this variant, we can interpret that this is a de novo variant and hence likely to be pathogenic. On the other hand, if one parent also has the variant which is present in the proband, then it is more likely to be non-pathogenic or less likely to be pathogenic because the parent is having the variant but he is not having the disease. However, we should keep incomplete penetrance in mind while making this interpretation. When we find a homozygous variant which is likely to be a VOUS, then again we need to do Sanger segregation analysis. So in Sanger segregation, if we find both parents are heterozygous, then it follows the typical Mendelian pattern and hence we will say this variant is likely to be pathogenic. However, if one parent is homozygous for the same variant as in the child, then we will say this is unlikely to be pathogenic because the parent is phenotypically normal. Sometimes one parent may be heterozygous and the other parent may be normal, and this you need to check the exon deletions. The large exon deletions can result in this kind of scenario where Sanger sequencing will not identify the presence of mutation in one of the parents. So Sanger segregation helps us to do the Mendelian segregation in the family, which helps in interpreting the variants. And the last evidence comes from the functional assay, which can be either a cell-based assay or a tissue-based studies or model organisms like Drosophila, zebrafish, and mouse. So this whole part forms part of the research laboratory. However, in a clinical setup, you could use functional assays like enzyme assay for lysosomal enzymes or hormone assays, or especially a reverse phenotyping, which can be very important in the clinic. So once you identify a particular variant, then you can use investigations like X-rays, MRI, CT scans to look for the classical features of that particular disease or clinical evaluation for dysmorphic features for that particular disease. So these are some of the examples of reverse phenotyping you can see here. If patient has Hunter syndrome, then the phenotype, we can look for the clinical and radiological features, and we can use a enzyme assay here to prove that this variant is disease causing or not. Similarly, if it is a calpainopathy and there are characteristic clinical findings, then we could use either muscle biopsy and immunoblot analysis to prove that the variant is disease causing. And similarly, if it is a PMP22 uh, involved in the neuropathy, then we could use the nerve conduction latencies to again prove that this variant is likely to be disease causing or not. However, once you collect all this evidence, just like a detective is going to collect all this evidence, but one evidence in its singularity cannot give us the final prediction. So we need to put all this evidence together and then the final judgment has to be given whether it is disease causing or not. So the guidelines for variant interpretation were given by the American College of Medical Genetics and Genomics along with the Association of Molecular Pathology in 2015. And these are the guidelines based on 28 criteria which are used world over to classify a variant. So based on all the evidences that we have talked just now, every variant is classified either as benign, likely benign, variant of uncertain significance, likely pathogenic, or pathogenic. So every variant has to be classified into one of these 5 categories. And for this, different kind of quantitative measures are used based on the evidences that we saw just now. So we don't have to go into the details of all these, but we should remember that the 28 criteria include the evidences that we discussed just now to classify it as benign or pathogenic. And you can have various different flowcharts to make things easier for us. So instead of going into these complicated pipelines, what we can do is that every variant we need to classify into one of these 5 categories. And for doing that, we have certain tools available. So one of the tools that is available freely is something called as VARSUM. So I encourage all of you to make use of this tool, which makes the same thing what we have discussed now. It makes it easier to understand. So this tool has a search bar here where you can enter your search in different formats, the particular mutation that is found in your patient. So once we can see, let us now see examples of how this VARSUM result comes out. So This particular variant now, CRTAP is the gene and this is the variant, C2T variant at this position. So it tells us about the variant here. This is a termination, so a stop codon variant. And the verdict given here is pathogenic. And below here you are getting what are the evidences based on which it is said as pathogenic. So you can see it is a nonsense variant. That's why it is likely to be pathogenic. Secondly, genome AD does not have, the count is 0. That means this variant is not reported in normal population, and multiple pathogenicity prediction software are predicting it as disease causing. So based on these three evidences, we classify it as pathogenic. Now we look at the next variant here. This is a variant in ARSA gene at this position. Now this variant is classified as likely pathogenic. So why is it classified as likely pathogenic? So here again we see the evidence. This variant is not found in the normal population, and there is a variant present at this particular location but with a different amino acid, and prediction software are predicting it as disease causing. So this variant but is a missense variant. The earlier variant was a nonsense variant. So here we do not have enough evidence to classify this variant as pathogenic. Hence, it is retained as likely pathogenic. The next variant, let us see here. So this variant is predicted as variant of uncertain significance. And why is that? Let us see. So this is a nonsense variant. So as I said, once you look at a nonsense variant, you should not always jump and say that this is causing. A nonsense variant also can be benign sometimes. So here you can see, although it is a nonsense variant, and one pathogenicity prediction software is classifying it as pathogenic, but ClinVar has classified this as benign in some of the patients. So there is conflicting evidence. Some evidence is there for pathogenicity, but some evidence is there for it to be benign, and hence it is retained as variant of uncertain significance. Now let us see this particular variant, which is classified as benign, and the reason is that this variant is not found in the normal population, but it is predicted as benign by the pathogenicity prediction software, and it is a synonymous variant. That means there is no change of amino acid. So because it is synonymous and predicted as benign, we are saying it as likely benign. Why we are not saying it as benign is because it is not present in the normal population. If the same variant was present in a large frequency in normal population, then we would have classified it as fully benign. And now let us see this final variant. This is classified as benign, and the reason is that it is present at very high frequency in normal population, and ClinVar classified it as benign, and pathogenicity prediction software also classified it as benign. So there is clear evidence that this is a benign variant, and hence it is classified as benign. So let us see in practice how we go about doing this. So this is the variant identified in one of the patients. So the patient has a variant in GUS-B gene, and the disease is mucopolysaccharidosis type 7, which is caused due to mutations in GUS-B gene. And you can see here, this is a missense variant here now. It is homozygous and it is classified as likely pathogenic. So how was this classified as likely pathogenic? Let us see one evidence at a time. So the first is the location based information. So here we can see this is a missense variant in exon 5 of GUS-B gene, and it results in amino acid change at this position. So this is a location or type or effect of the variant. The next evidence is that this variant has not been reported as normal variation in various population databases. So we think that this variant is likely to be disease causing. So this is the normal population database evidence. Then this variant has not been reported in other patients with mucopolysaccharidosis type 7 in OMIM ClinVar. So this is the known variant database evidence. Then the fourth is in silico pathogenicity prediction software like CAD is giving a score of 24.8, and mutation tester has predicted it as disease causing. So this is a prediction software based evidence. And then next, very important, is the clinical features of this patient are matching with mucopolysaccharidosis type 7. Hence, based on these all evidences, so this is the most important, the genotype phenotype correlation based evidence. So This is the most important evidence that we use in absence of a functional assay. We can look at the genotype and phenotype match, and if it is matching, then it helps us to interpret the variant. So in this way, looking at all these evidences, we can classify this variant as likely pathogenic variant. Now, why we are not saying this variant as pathogenic variant is because it has not been reported earlier in patients with mucosal polysaccharidosis. If it was reported earlier, then we would have classified it as pathogenic. But now, since it is a novel variant, but it has all the characteristics of a disease-causing variant, we are classifying it as a likely pathogenic variant. So, in this way, we can easily classify any variant by taking the stepwise approach using location-based classification, then looking at the frequency in the normal population, then looking whether this is a known variant in the databases, then looking at the pathogenicity prediction of this variant, followed by Mendelian segregation in the family, and then most important is the genotype-phenotype correlation of this variant with the known clinical phenotype. Now, what do you do when you get a VOUS? So when you get a VOUS that is reported in the exome sequencing, the first step is that you can do the variant interpretation yourself and compare it with what the particular lab has reported. Secondly, very important thing that a clinician can do is that you try to do reverse phenotyping, that is match the clinical phenotype with the VOUS and see whether the matching phenotype is present. Thirdly, Mendelian segregation in the family can sometimes help us to reclassify the variant, and you should check if a functional assay like an enzyme assay or any other investigation is available. However, if you are not able to classify a variant, then follow up the patient periodically and check for reclassification of the variant. So this classification will keep on changing as more and more evidence is available in different databases. However, very important thing to remember is do not take up any irreversible actions like doing prenatal diagnosis or therapy based on a VOUS. A VOUS or a variant of uncertain significance should be retained as a variant of uncertain significance, and any irreversible action like prenatal diagnosis or treatment of the disorder should not be done based on this kind of variants. So in summary, proper interpretation of variant is far more important than finding the variant, and knowledge is changing at a fast pace, and hence clinicians need to be aware of the recent information, and fallacies of the variant interpretation need to be kept in mind to avoid making diagnosis and counseling mistakes. And clinical correlation of the variant and reverse phenotyping is very important in this era of high throughput genomic assays. So it is very important that the clinicians are able to interpret the variant themselves and not rely on the particular laboratory who is giving the interpretation. So as I said before, by following the simple steps one after another, anybody should be able to do the variant interpretation. So I hope all of you are confident now to do variant interpretation. So go ahead and take up the reports that are given to you by the laboratory and do the variant interpretation yourself. And now you can try and answer the questions that are following. And if you have